All right, good evening, everyone. It's a pleasure to welcome you all tonight, and an even greater pleasure to introduce Dr. Gergely Toth. Dr. Toth is not just the visionary founder of Cantabio Pharmaceuticals, which is a pharmaceutical company at the forefront of developing precision therapeutics and diagnostics for neurodegenerative disease, but also an expert with over 2 decades of dedicated research into Alzheimer's and Parkinson's disease. So Dr. Gergely Toth embarked his journey by earning his PhD at Creighton University in the US in 2001. His quest for knowledge then took him into the halls of UC Berkeley, where he refined his research as a postdoctoral fellow in the Department of Molecular Biology. Gergely then worked at the innovative biotech company Protein Mechanics, which was sold to Locus Pharmaceuticals, before leading a team at Elan Pharmaceuticals working on Alzheimer's disease. From 2010 to 2015, Dr. Toth sharpened his expertise via a Wellcome Trust research fellowship at the University of Cambridge. So today he continues to share his valuable and pioneering insights to the academic and scientific communities as an honorary associate professor at UCL. So ladies and gentlemen, please give a warm round of applause to welcome Dr. Gergely Toth.
Thank you very much for this nice introduction, and thanks everyone for coming. I can see it's a hardcore Parkinson's disease therapeutic diagnostics interest audience I have here, so thanks. And mostly I want to thank obviously the Cambridge Longevity and Aging Society for inviting me to talk about our research at Cantabio Pharmaceuticals, really targeting DJ-1. With that, let me jump right in and talk a bit, really start very far away, just to give a bit of perspective of our vision with our therapeutic approaches targeting DJ-1 and developing a disease-modifying therapeutic for Parkinson's disease. So we are very much interested in redox homeostasis, and I think most of you know what redox homeostasis is, but in fact, as we talk a lot about oxidative stress and its essentially negative role in the onset of diseases, but in fact, oxidative stress comes from essentially reactive oxygen species, which are fundamentally very much required messengers that actually control a lot of basic fundamental processes in cells. And these reactive oxygen species, when they are at low concentrations, they are imbalanced with kind of antioxidant enzyme systems in cells. And then we get redox cellular homeostasis and everything works fine. Now, as we age, or perhaps due to environmental factors, lifestyle factors, or disease, this balance essentially comes out of balance and results in cellular dysfunction, damage, and cell death. To look into this a bit more, we can look under the hood what's happening, right? So we got various reactive oxygen species. Think about hydrogen peroxide, superoxide, nitrite oxide, and so on. They're actually sourced from mitochondria and from various other enzymatic systems. They're controlled by, again, antioxidant systems. Now, at low exposure or at low concentrations, they actually control various redox signaling networks. And again, if everything is in order, then we got normal function. However, when this is disrupted, we have oxidative stress or oxidative distress, and various things go wrong. Now, again, Ross has a number of positive functions, right? So stem cell renewal, proliferation, healthy immune response, and even longevity. On the other side, if those levels are high, then we get stem cell exhaustion, tumorigenesis, autoimmunity, senescence. And in fact, what we are really interested in is neurodegeneration. And in fact, in Parkinson's, it is well known that oxidative stress is a very important, critical process that drives disease onset. So why is that? Well, there are certain genetic defects, certain mutations in certain genes that actually end up increasing oxidative stress through various mechanisms, environmental toxins, and of course aging. All can result in various biological processes that increase reactive oxidative species. Okay, so for example, one of the most important one in Parkinson's is actually mitochondrial dysfunction. Mitochondrial dysfunction results in high oxidative stress. Another thing that's really specific to Parkinson's disease is actually dopamine metabolism and dopamine oxidation due to high oxidative stress. Now, high oxidative stress in neurons are particularly vulnerable to oxidative stress. And in different ways, it leads to neurodegeneration. I'll give you two examples. One is protein homeostasis. In Parkinson's, actually, synuclein oxidation drives synuclein oligomerization and Lewy body formation. Or actually, self-defense, essentially anti-stress defense, is also damaged by oxidative stress. Okay, so these are all specific to Parkinson's. Sorry, I have a problem with accessing. Now, to dive into this a bit more, in redox signaling, there are certain proteins that are responsible for redox signaling through reacting with reactive oxidative species. In these proteins, the function of these proteins are actually controlled by reversible oxidation. I'll talk more about that. Some of these proteins are oxidoreductases, transferases, hydrolases, lysases, isomerases, and ligases. So there's actually a fairly high number of proteins that are redox activated or redox controlled proteins. The way this actually happens is quite complicated, and I don't have time to go into that, but a lot of these proteins contain one specific cysteine, which is highly reactive, mostly due to the fact that they are probably arranged in three dimension in the structure of these proteins, where they are actually in a thiolate form, which means that they are super reactive. And so these cysteines can actually be modified and oxidized in various ways, very sensitively. Now, when oxidative stress increases, as you can see here in the red, these cysteines can actually oxidize, forming sulfonic acid and sulfonic acid. And some of this sulfonic acid is actually an irreversible and essentially inactive form, where sulfonic acid can be essentially actually reversed. And so all these modifications have a very particular function in redox signaling. Now, in oxidative stress and high oxidative stress, a lot of times what happens is these superreactive superreactive cysteines actually overoxidize, get inactivated, and essentially these enzymes are not functional anymore. And so oxidative stress, a lot of times, and high oxidative stress, the main damage that it does to cells is not necessarily a structural damage where it reacts randomly with different side chains, causing essentially protein structural destabilization, but rather very specifically knocking out these redox signaling enzymes. And essentially that's how the majority of the damage of oxidative stress happens. Now, this is kind of the background where we start from. At Cantabio Pharmaceuticals, our vision was, okay, well, there are these essentially processes like oxidative stress that really cause damage to cellular function. But in fact, it's not just oxidative stress. There's other reactive stresses, for example, glyoxal stress, and indirectly, for example, even protein aggregation that causes oxidative stress, that causes damage. And SVH, these actually build up and can cause the onset and progression of a number of diseases, for example, Parkinson's and Alzheimer's disease. And so we are very interested in reducing these stresses. The question is, what essentially have been done was to reduce these stresses in a focused way. So oxidative stress, we need antioxidants. Protein aggregation, we need aggregation inhibitors. Now, this is great, but the problem is that these stresses are very connected. So oxidative stress increases protein aggregation causes oxidative stress, glyoxal stress causes oxidative stress, inflammation, which causes more oxidative stress. So what we need approaches where we actually have a systematic response to reducing these type of stresses, which is actually not that easy. Now, it happens that we have a target, DJ-1, which I'll talk a lot about, which actually can maybe be modulated in a way to counteract these stresses because DJ-1 is a controller of redox and protein homeostasis. And I'll explain what I mean by that. But before I do, let's look at kind of Parkinson's and Alzheimer's and the pathology of it. It turns out that let's say biochemical stress is very much a main pathology of these diseases. Think about Alzheimer's disease. Obviously, everybody has heard of amyloid and amyloid buildup. Tau protein aggregation results in neurofibrillary tangles. Alpha-synuclein aggregation results and collects in Lewy bodies, and so on and so on. And so we know that, and we can detect that, for example, with very specific PET imaging agents. And from these, and now with some breakthroughs in Alzheimer's disease in CSF, essentially IVDs, looking at essentially biomarkers of let's say tau phosphorylation or A-beta levels, we can actually see and diagnose these pathologies. And so what we learned from all of this is that these pathologies actually are present way before the symptoms arrives, possibly 10, 15, 20 years before. So if you can see in this diagram, you got a clinical disease stage. So from here you have symptoms, and essentially both A-beta and tau aggregation and pathology, fibrillary deposition actually happens way before that. And neurodegeneration is happening. If you look at oxidative stress and glyoxal stress, it's actually very similar. These oxidative stress and glyoxal stress is very apparent very early on. The challenge is that we don't really have a very good ways to measure actually both glyoxal stress and oxidative stress in the brain at this point. So that's a challenge. But from post-mortem brain examinations, we know that there's a lot of lipid peroxidation, advanced glycylation, and products in both diseases. Let me switch to DJ-1 and why we are working on DJ-1. As I mentioned, DJ-1 is a control of reduction protein homeostasis. And one of the reasons why we started working on DJ-1 is because it's also a genetically validated target, means that mutations in human in DJ-1 end up causing familial Parkinson's disease. Very simply, if someone has a very specific mutations that causes loss of structure, loss of function of DJ-1, he or she will get Parkinson's, unfortunately. Okay, so that's a very strong validation that DJ-1 is likely involved not just in familial Parkinson's, but actually in idiopathic Parkinson's disease. Now, DJ-1 is a protein, it's a homodimer. It's actually a small protein, and you can crystallize it, and you can work with it from a biophysical and structural point of view. So it's very amenable for small molecule drug discovery and all kinds of drug discoveries. So that was a plus factor for a small company like us to really try to tackle this. One of the caveats, however, is DJ-1 is a activated redox-controlled protein. It has a very reactive cysteine that's controlled by oxidative stress. And in fact, there are essentially no drugs out there that that control and target proteins that are kind of redox, is involved in redox signaling, that can properly modulate this redox modulation of these proteins. So it's a very novel target from a therapeutic point of view. So what does DJ-1 do? Well, when it's synthesized or expressed, it has a biochemical function. It's also a very strong chaperone. So it has an enzymatic function, which I'll talk about. It's also a chaperone. It's a transcriptional factor. It's actually expressed inherently in mitochondria. It enables mitochondrial function. And it's a transcription regulator. What happens is when there's high oxidative stress, we believe DJ-1 oxidizes, locates in the nucleus, and initiates an anti-stress response by overexpressing proteins that can counteract oxidative stress, and also chaperones that limit protein aggregation. So it does a lot of things, and in fact, it's also involved in signal transduction, enabling cell survival pathways to be activated. So a lot of times when I talk to, for example, investors, they also say, well, what does DJ-1 do? And I say, well, it does all of these things. That's the advantage of it. It does everything just a bit, and it adds up, and it's a very powerful way to possibly counteract a very complex disease. And so that's an advantage and a disadvantage, obviously, from a drug discovery point of view. Now, because of that, it's relevant to a number of oxidative stress connected diseases, obviously Parkinson's, diabetes, stroke, and even male infertility. And in these diseases, there's a loss of function. So from a therapeutic point of view, we want to increase its activity. While in cancer, tumor cells figured out that DJ-1 is a cell survival protein, helps cells survive, so they actually increase its level and its activity. So in cancer, we want to knock it out and inhibit it. Totally the opposite. Let me dive into a bit more about how DJ-1 is actually controlled by oxidative stress. So DJ-1 has a very reactive cysteine, which is cysteine 106. It's in a tylate form. It can easily oxidize into sulfuric acid form, which I'm going to call this oxidized DJ-1 in this lecture. And then at prolonged high oxidative stress levels, it can overoxidize into sulfonic acid form, which I will call overoxidized DJ-1. We know that this is enzymatically active. We know this is enzymatically inactive. I'll show you some data on this. This likely has an effect in neuroprotection due to high oxidative stress. And this is a pathological state because this is an unstable state, in fact. Now, we make DJ-1 in the lab, right, recombinantly, and we can actually oxidize, reduce DJ-1. Here, as I show here, this is basically mass spec. It's a proteomics study where we're looking at following oxidized and overoxidized states. So we can oxidize by adding more hydrogen peroxide. We actually can fully oxidize DJ-1. And when we add high levels of hydrogen peroxide, the overoxidized state will actually appear. So we studied in detail, from a drug discovery point of view, we really wanted to understand the structure. We really do a lot of structure-based drug design, so we wanted to understand the structure, and that's fundamentally also very important from signaling point of view and protein-protein interaction point of view. And what's interesting is that if you look at we crystallize both the reduced and oxidized DJ-1 in basically the same conditions, and what we found that the structure of reduced and oxidized DJ-1 is basically exactly the same. So that was interesting because, okay, if the structure is the same, how can they have different activities? And essentially, how can they localize in different compartments of the cell differently? But when we looked at it closely, we could see that there are small differences in the active site. And so maybe that's the difference. Another thing that's really interesting is that oxidized DJ-1 thermally is much more stable. So the whole homodimer itself is more stable compared to reduced DJ-1. We also seen that overoxidized DJ-1 we showed with biophysical methodologies that it actually is very prone to aggregation. It's unstable. And we've done molecular dynamic simulations where we looked at essentially calculated the free energy of the complex for reduced oxidized and over-oxidized. And what we could see is that actually over-oxidized is less stable. And most of this due to the fact that the third oxygen somehow destabilizes the homodimeric interface. So let me kind of switch gears and talk about what happens to DJ-1 in Parkinson's disease, because that's really important to understand in order for us to actually target it therapeutically. So what can we learn from essentially human mutations? One of the things that we know that most of the human mutations actually, again, cause structural instability, or actually the protein never folds. So there's no active DJ-1. So in this case, for example, a study on a patient with DJ-1L172Q mutations showed that there's no staining for DJ-1 in postmortem brain tissue. So there's no active DJ-1. The second thing that we know is that actually there's very strong Lewy body pathology with DJ-1 mutant patients. Okay, so synuclein is aggregating, and so that's very, very important also, because most of, let's say, Parkinson's have synuclein pathology, but not necessarily, all patients. Another patient study showed, this is a T154K mutation, that there's actually mitochondrial dysfunction and high oxidative stress. And no DJ-1 can be measured in a blood test by ELISA. Okay, so in summary, what we know from these is that we have essentially high synuclein pathology and mitochondrial dysfunction, high oxidative stress due to DJ-1. Kind of goes back to all the basic functions that I've been telling you. Now, in idiopathic patients, it's less unclear what's happening, right? Because it's much harder to essentially tell. But we know from one really well done study is that actually in the brain, in postmortem brain tissue, there's a significant loss of DJ-1, of total DJ-1. The second study by a Japanese group showed that in erythrocytes, in blood, there's actually more oxidized DJ-1, cysteine 106 oxidized DJ-1. And that's actually very significant, although this is a small study. So this is what we know from human studies. Now, we've been working with DJ-1 for 10 years, and we do a lot of biophysical, biochemical studies. And as some of my colleagues were working with DJ-1, they noticed that this damn protein tends to aggregate, and it's hard to work with sometimes. And so we started looking at this, and a study came out of this, which we published recently, in which we showed that actually DJ-1 can misfold, oligomerize, and actually form these fibrin-like insoluble deposits. And when we looked at this in more detail, we saw these are fibrin-like, and actually, based on this, we actually looked at what could be happening in postmortem brain tissue, which I'll talk to you in a minute. So first, we just done very basic things where we say, okay, well, are we losing soluble DJ-1? And so we can't quantify soluble DJ-1 as a function of time, and we saw that we were losing some soluble reduced DJ-1, but actually we were losing a lot more oxidized and over-oxidized DJ-1. So DJ-1 seemed to aggregate. Okay, so what kind of aggregates are we getting? Well, it turns out that these are thioflavin T positive aggregates, and most of you know that thioflavin T stands for beta sheet-like structures. For example, amyloid neurofibrillary tangles also are identified with thioflavin T. And then we used FTR spectroscopy, and we showed that actually there's high as DJ-1 aggregates, there's a significant increase in beta sheet structure, both for reduced oxidized and over-oxidized. CD studies essentially supported this, where we used ultracentrifugation, looked at the palate and the supernatant, and we saw that the palates are fully beta sheet-like. So all of this suggested that DJ-1 actually, in fairly close to physiological conditions, can form thioflavin T positive fibrin-like structures. And we said, wow, okay, we thought this was a very stable protein. How's this possible? But okay, maybe this is at fairly high concentrations. and this is an artifact. Well, let's look at the brain. And so in collaboration with Clinical Neuroscience Department, Roger Barker and his team and Frank and Albiro, we looked at Parkinson's and Alzheimer's brain tissue slices. And here we stained for fibrils using PFTA, so you get the green fluorescence. We used an oxidized DJ-1 antibody. We stained for nucleus. And so if you kind of look at this, the merging of this, you can see that you got stronger staining for oxidized DJ-1 that co-localizes with fibril, these PFTA staining, suggesting that where there's a lot of fibril structures, there's also oxidized DJ-1, and none of this happens essentially in control. Okay, well, we looked at phosphoserine, meaning, well, Lewy bodies, right? And we saw, interestingly, that there's actually co-staining also, but it's essentially DJ-1 is kind of oxidized DJ-1 is around these phosphosynucleins, so it's not really co-localizing, but likely in Lewy bodies around synuclein. And what was the most interesting, that actually oxidized DJ-1 really co-localized with phospho-tau, essentially neurofibrillary tangles. And we've seen similar things in Alzheimer's disease for tau, and in fact, we saw some co-localization with amyloid. Perhaps this is not that surprising, because actually in amyloid, in Lewy bodies, there are other 400 proteins. We know that these are not clean fibril structures, but there's a lot of other proteins that co-aggregate with this, and probably DJ-1 also co-aggregates. So really the question now, is this a co-aggregation, but actually does DJ-1 actually aggregate by itself? So this is something that we are very interested in exploring, and we're doing some work on this. We don't have the answer yet. But one thing, just to kind of summarize, what's really important for us from these postmortem studies, we also can do a semi-quantitative analysis which suggests that actually there are increased oxidized DJ levels in Parkinson's and Alzheimer's disease. And that's actually very important, kind of going back to redox homeostasis idea. Okay, so that's it. And if you look back to the literature, there's a lot of hints about DJ-1 aggregation, but no one has actually taken and explored it in as much detail as we have. So DJ-1 has been observed to present in high molecular aggregates in Parkinson brain tissue. It's also in urosoluble fractions in Parkinson's brain tissue. Actually, there's a Korean group that a long time ago published that actually DJ-1 aggregates, but their aggregates were not fibril necessarily, but they suggested that DJ-1 keeps its structure. And then there's been a number of publications showing that essentially total DJ-1 and synuclein co-localizes and tau co-localizes. So there's a lot of hints really supporting kind of our conclusion that actually DJ-1 aggregates and oxidizes, and this is very much part of the pathology in Parkinson's disease. Okay, so I mentioned that DJ-1 also has a chaperone function. And in fact, for over 10 years now, there's been several studies that showed that DJ-1 can inhibit alpha-synuclein aggregation. And in fact, there's one or two studies that actually showed that only oxidized DJ-1 inhibits aggregation. So that's, again, a very interesting phenomena. Reduced DJ-1 doesn't seem to interact with DJ-1. So we were very interested in understanding this in more detail. And so we did the following study. We showed that actually looked at the interaction between monomeric synuclein and DJ-1, and we found that only oxidized DJ-1 interacts. And using MR spectroscopy, we identified that actually the N-terminal of alpha-synuclein interacts with DJ-1. And this is actually a quite transient interaction in NMR, but when you do the biophysics, this is a potent interaction. So the KD, we use 3 different methodologies to show that the KD is under 100 nanomolar. This is a quite strong interaction for an intracellular protein. In fact, we have, so monomeric DJ-1 and oxidized DJ-1, so monomeric alpha-synuclein and oxidized DJ-1 interact. But what happens in an experiment when both proteins are in aggregating conditions. And so we use dynamic light scattering experiments to probe that. With dynamic light scattering, we can really probe and measure early oligomer formation. And from all this, what we've seen that oxidized DJ-1 alpha-synuclein can essentially inhibit oligomer formation. And oligomers only start appearing after 70 hours that they are in these conditions. In this condition, only at 70 hours, we starting to see oligomers. With reduced DJ-1 and alpha-synuclein and over-oxidized DJ-1, we actually see aggregates appear in the first few hours. So this is a massive delay of oligomer formation and inhibition of oligomer formation. So this is a very, very interesting system, and suggests basically that when oxidative stress increases, DJ-1 oxidizes, and in these disease conditions, when alpha-synuclein is more likely to aggregate, that's only when these two proteins may actually meet in the cell or neurons and result in essentially a stabilization and reduction of alpha-synuclein aggregation. But if we go further, but actually this is a very unique case, because we're not just talking about synuclein aggregation here, but we're talking about DJ-1 aggregation. And when you put this together, neither synuclein nor DJ-1 actually aggregate. So this is beneficial for both protein systems. Let me switch gears again and talk a bit about DJ-1 enzymatic function. So DJ-1 has a number of enzymatic functions. So it's a weak protease. It's actually quite strong esterase, and it's probably a fairly weak glyoxylase. But its glyoxylase function is actually also very important, because essentially glyoxylase is really part of the pathology in Parkinson's and Alzheimer's. So what is glyoxyl stress? This is not so much in the mainstream, right? So glyoxyl stress is basically glyoxyls are reactive aldehydes, right? That can just interact randomly with protein side chains and cause, oxidative stress, inflammation, and hypoxia. They actually cause advanced glycylation end products. Advanced glycylation end products generally end up causing inflammation and then oxidative stress. Now, methylglyoxal is one of the most abundant glyoxyls in our body, and most of methylglyoxal is actually a byproduct of glucose metabolism. So if you've got high glucose levels, you're going to have higher glyoxal stress. And actually, glyoxal stress is very important in diabetes too, and vascular complications in diabetes. Now, in Parkinson's, again, this hasn't been mainstream, but in the last few years, there have been some really good work showing the pathology of advanced glycylation and products in Parkinson's brain. And also have been shown that methylglyoxal reacts with alpha-synuclein and causes its oligomerization, and causes toxic oligomer formation. And DJ-1 can degrade methylglyoxal and other glyoxyls and actually form lactate. Lactate is generally protective at low concentrations. Now, we've been very interested in glyoxylase because we said, okay, well, DJ-1 does a lot of things. Maybe this is the main function of DJ-1. And so we've been looking at this from various angles. One way obviously we are a pharmaceutical company, so we developed some inhibitors, some potent specific inhibitors. And actually we developed covalent inhibitors. One of our novel covalent inhibitors inhibits glyoxal activity at about 900 nanomolar, which is not super reactive, but it's enough as a tool compound to look at a couple of things. And so one of the things that we've done with this inhibitor is we tested it in a glyoxal toxicity assay. And in here, what we're doing is you're adding glyoxal to cells. These are neuroblastoma cells. We have wild type in red. We have knockout in green. And then we have overexpressed B3 and F4 is actually DJ-1 overexpressed thing in these cell lines. And what we can see is there's a huge difference between the cell viability and essentially the cell's resistance to glyoxal stress. DJ-1 overexpressing cells can really essentially are less sensitive to glyoxal stress and then wild type and knockout. And knockout is just a little bit more sensitive compared to normal cells. And then we can take our inhibitor, and what's really interesting is these are essentially, we basically use one concentration of glyoxal. This is actually 4 millimolar here. And when we treat with our inhibitor, we basically knock out DJ-1. You can see that the cell zone, depend on the doses of inhibitor, we see essentially cell viability goes down. So as we knock out DJ-1 with our covalent inhibitor, the cell zone essential ability or defense against glyoxal stress really goes down, and therefore ends up essentially in cell death. Now, obviously, this type of therapy wouldn't be good for Parkinson's, because you would essentially cause Parkinson's. However, this could be very good if it's very specific against cancer, because we want to knock out DJ-1, because DJ-1 is involved in essentially cancer onset and proliferation in many different ways. I'm not going to go into that today. I'm going to focus on Parkinson's and neurodegeneration. So another another thing that we've really pioneered and looked at is, okay, well, what happens if we oxidize DJ-1? Well, in fact, when we oxidize DJ-1, we lose enzymatic activity, which is not a surprise, because we modify the catalytic active site of DJ-1 and it's no longer able to do its enzymatic function. But this is really interesting, because I've been telling you that actually this is a normal function of DJ-1 to get oxidized at lower levels of hydrogen peroxide. Maybe it picks up new functions, but it loses its enzymatic function. And in fact, essentially oxidation at cis-106 completely changes the function of DJ-1, we believe. And also, as I mentioned, location and localization. Okay, so just focus on Parkinson's. Actually, there's a number of other metabolites and things that interact with DJ-1 at cysteine-106. For example, in Parkinson's, Parkinson's obviously is the degeneration of the substantia nigra, the dopaminergic system, where dopamine is generated, right? So what happens is, when you generate dopamine can react very easily with hydrogen peroxide and oxidative stress to cause these very reactive dopamine quinones. And in fact, probably that's why oxidative stress is very specifically relevant to the dopaminergic system, because if you increase oxidative stress, you got dopamine oxidation. Dopamine oxidation causes toxicity, mitochondrial dysfunction, and causes more oxidative stress. So it's a vicious cycle. Anyhow, so interestingly, oxidized dopamine can react with cysteine-106 and inhibit DJ-1. And it's not just inhibits DJ-1. If you look at the thermal stability of dopamine-reacted DJ-1, actually there's a loss of structural stability and thermal stability. And actually, oxidized dopamine-modified DJ-1 is very prone to aggregation, but it does not cause fibril aggregates. It just causes amorphous aggregates. You still lose your structure. You still lose your DJ-1 activity. So again, this is actually a very specific. So oxidized dopamine-modified DJ-1 is a very specific Parkinson's relevant product, and it probably would be a great biomarker if you could measure it easily. So this is something that we are very interested in also, obviously, looking at. Just to kind of finish my introduction, there's a number of studies that have been shown in vivo in mice and rats that DJ-1 is a good target for Parkinson's. I'm not going to go into that, but I guess one important thing is that you knock it out in mice, and mostly in rats, you get Parkinson's-like pathology and symptoms. And if you overexpress it, then it's protective in a number of Parkinson's models. Now, at Cantabio Pharmaceuticals, we are focused on DJ-1 very, very much. And we have small molecule programs, we have enzyme replacement programs, we have a platform where we can measure DJ-1 and oxidize DJ-1 very precisely in blood and tissue lysates. And so I'll talk a bit about some of the applications here and therapeutic approaches that we are doing. I also mentioned that we also are very interested in protein misfolding and have our own approach there, which I won't talk about today. But actually, a lot of DJ-1 work on oxidative stress and protein aggregation is very much linked to tau, synuclein, and A beta. And so these projects are actually, this research is very much interconnected. Okay, so one of the things we're doing, as I mentioned, there's high oxidative stress. DJ-1 is more likely to lose its structure. And so one of the things that we've been working on is developing these, what we call pharmacological chaperones. Very simply, compounds that bind to DJ-1 and enhance its structure. And in fact, this translates to longer lifetime in the cell, longer activity. This is actually a very complicated mechanism. Nevertheless, what we did here was to say, well, let's look at this the simplest way. Let's get reduced DJ-1, oxidized DJ-1, look for binders of small molecules. And we did this with a screen called chemical microarray screening. And idea is that you got a chemical microarray, small molecules are immobilized. You got DJ-1 added to it, and when there's a binding event, you detect that with surface plasmon resonance. So we worked with Graffinity Pharmaceuticals with this and screened 110,000 compounds, got a bunch of hits, very different scaffolds. I'm not going to go into this. And just to say that we pursued a number of small molecules here, and we developed one compound, one scaffold in more. This is called CB125 that interacts with DJ-1 potently, KD about 200, 300 nanomolar. It stabilizes the protein. It's an allosteric side binder. It doesn't bind to and doesn't inhibit DJ-1 enzymatic functions. And then, interestingly, it has various and fairly high activity in Parkinson's toxin models. So, for example, in an MP cell model, an EC50 is about 220 nanomolar. But it's also relevant in a paracut cell model. These are all mitochondrial dysfunction, high oxidative stress, Parkinson's models. And actually, interestingly, it's also active in a proteasome inhibition cell toxicity model. So with this, we went further, and when we were developing, we actually developed a brain-penetrant, orally bioavailable compound. And this was active in an MPTP mice model. I'm probably going to just cruise through this just to say that, in fact, this compound was very active, meaning that when we treated MPTP, mice with MPTP, there's a loss of essentially dopaminergic neurons stained with TH plus neurons, and this compound can completely alleviate that, and also essentially alleviate some dopaminergic loss and full dopac loss. So this is very promising. I'm going to be honest, we still have a long way to go to understand its full mechanism, but this is something that obviously it works, and we are really pushing it forward. Another therapeutic approach is more straightforward, but equally challenging due to other things. Well, as I said, in Parkinson's, we want to increase DJ-1 activity. How can we do that? Well, we can just inject more DJ-1, possibly, or somehow express DJ-1 in vivo. Now, obviously, this is not so simple because DJ-1 is a intracellular protein. So when you inject it, it doesn't cross the membrane. So what we have done, and this is not our idea, people are doing this, we created a construct where we added a cell-penetrant peptide to its N-terminal. So you got the homodimer here, gray is the cell-penetrating peptide. And in fact, luckily, this actually takes it and gets it into the cell. And actually, its cell penetration is quite efficient. Now, this doesn't work with every protein. It works with small proteins, obviously, and other peptides. With larger, more complex proteins, this approach doesn't work. So just to say is that what's really nice about our drug candidate is that it has exactly the same structure as native DJ-1. We crystallized both, overlaid it. It's exactly the same. And in fact, we've done some local dynamic simulation some time ago on this. And why this is interesting is because essentially the idea is, okay, does the cell-penetrating peptide part, the yellow part, does that interfere with the active site? Does this interfere with any protein-protein interaction site? And we think it doesn't. This is a microdynamic simulation, and you can see that essentially the cell-penetrating peptide is actually very flexible. Here, this is the active site here, and it does not interfere with any of the main binding sites, we believe. So that's a good sign. Now we looked at its cell penetration in different cell models. So this is a DJ-1 knockout cell model. These are HEC293 cells, and we administered to cells DJ-1 as cell penetrating DJ-1, used immunofluorescence. And in DJ-1 you can see some background. This is just sticking to the plate, while here we can actually see cell penetrating DJ-1 actually getting into cells. And we used our Redux DJ-1 platform to really characterize its concentration and kinetics. And so when we add cell penetrating DJ-1, very quickly we see high levels of cell penetrating DJ-1, and as in 24 hours that actually reduces. And that makes sense because cell penetrating DJ-1 will get degraded. Half-life of DJ-1 is about 24 hours in the cell. When we do DJ-1, we have no DJ-1 in the cell, so it does not get no DJ-1 gets into cell penetrating DJ-1 gets in cell. So we have also looked at this in several Parkinson's models. So one is a paraquat model. This is again a mitochondrial dysfunction model. So cell penetrating DJ-1 is protective here. And actually one of the most interesting models and simplest models is actually a glyoxal stress model there. We add methylglyoxal to cells. We completely kill the cells. But if we pre-treat with cell-penetrating DJ-1, we can essentially rescue cell viability loss. Simply, we degrade methylglyoxal into lactate and essentially reduce toxic glyoxal levels. However, again, with oxidative stress and glyoxal stress is really not necessarily that mainstream Parkinson's research. It's all about synuclein aggregation, as you know. So everybody wants to see alleviation of synuclein aggregation of drug candidates as a disease-modifying therapeutic. And in fact, as I mentioned, DJ-1 is a good chaperone inhibitor of alpha-synuclein aggregation. And in fact, this cell-penetrating DJ-1 is an awesome inhibitor of alpha-synuclein aggregation. So we have alpha-synuclein at 100 micromolar, right? In this assay, we got the blue is showing tau-flavin T, fluorescence indicating that synuclein is aggregating. And when we add cell-penetrating DJ-1, we completely inhibit aggregation, and we can inhibit already at 10 micromolar. So substoichiometrically, 10%, we can go down substoichiometrically 10% of DJ-1 will inhibit alpha-synuclein aggregation. And it makes sense because it stops nucleation. It stops nucleation of, and so very low levels can have a drastic effect at initiating, just postponing nucleation events in synuclein. So we are very excited about this and really kind of focusing on essentially a protein inhibition strategy with cell-penetrating DJ-1 for Parkinson's. Okay, so I'm getting to the last thing that I want to talk about at this point is the fact that, okay, well, we're building these therapeutic projects, but in fact, it's very important to measure DJ-1 levels and oxidize DJ-1 levels because it helps to understand, we need to understand mechanism. We need to understand whether some of our drugs candidates are targeting DJ-1 in vivo. And so we started building various methodologies to quantify DJ-1 because these are not available. You can buy a total DJ-1 ELISA kit, but you cannot buy an oxidized or over-oxidized or reduced DJ-1 ELISA kit. So we developed conformational specific oxidized state specific antibodies and use for essentially reduced DJ-1, oxidized DJ-1, and over-oxidized DJ-1. Let me correct myself. We couldn't get antibodies for reduced DJ-1, so we designed our covalent inhibitor to be a molecular probe, and we built these sandwich ELISAs with this. And the idea here is that in healthy cells, in healthy people, you have mostly reduced DJ-1, maybe a bit of oxidized DJ-1. But as disease comes along, you have high oxidative stress, DJ-1 gets oxidized. So you get less reduced DJ-1 and more oxidized DJ-1. Pretty simple. And perhaps this oxidation will actually increase as disease increases. And so we have this technology working, so we call this Redox DJ-1 ELISA platform. And so we have a couple of validations here. So these are cell models. So these are two cell models. One is where we add hydrogen peroxide to neuroblastoma cells, or we add 6-hydroxydopamine. Both are basically very simple Parkinson's models, cell models. And so we look at total DJ-1 with this ELISA system. Green is control, red is 6-hydroxydopamine treated cells. You can see that in this model there's a loss of total DJ-1, which is quite interesting. That actually recapitulates, what we see, what people, UCL researchers saw in sporadic Parkinson's, meaning there's loss of total DJ-1 levels in postmortem brain tissue. When we add hydrogen peroxide, we actually don't really change total DJ-1 levels. And then these are DJ-1 cow levels. So essentially, when we knock DJ-1 out, so this is basically kind of the background here. Okay, so let's see what happens to oxidized DJ-1, reduced DJ-1. Well, in oxidized DJ-1 levels will go up in a bit with 6-hydroxydopamine treatment. That also makes sense because essentially you got oxidative stress generated by mitochondrial dysfunction as 6-hydroxydopamine ends up causing high oxidative stress, and essentially hydrogen peroxide addition, of course, will increase oxidized DJ-1, and exactly the opposite happens in reduced DJ-1. So you got loss of reduced DJ-1 levels in 6-hydroxydopamine and loss of reduced DJ-1 in hydrogen peroxide levels. So this is great because fundamentally what we want to see, we kind of see this in cell models. So what we're doing now is we're looking at postmortem brain tissue. So we've looked at healthy brain tissue, and what we see is that there are total DJ-1 levels, and essentially there's a little bit of oxidized DJ-1 and there's a lot of reduced DJ-1. We also looked at, now we're really focused on a blood test. So we're looking at plasma with this. And so what we can see that total DJ-1 levels in healthy volunteers, are vary quite a bit. Oxidized DJ levels are a bit more constant, and also reduced DJ-1 levels vary quite a bit. And then what we can see if you look at the total levels, that reduced and oxidized and reduced kind of equals total levels, which also makes sense. And what we're trying to do with this is why this is exciting, because we like to think of this as a potential precision diagnostics. Why is that? So we are looking at the ratio between oxidized and reduced. And we can also control this with total DJ-1. So there's a lot of difference between age. Younger people will have less DJ-1 or possibly more DJ-1. We don't know. We haven't looked at this. Older people may have more oxidized DJ-1. So their total DJ-1 levels could change dependent on age and sex, right? So with being able to control with this is an advantage. And so again, in the future, what we're doing, we're looking at Parkinson's postmortem brain tissue and essentially Parkinson's patient's plasma in big studies to essentially do proof of concept studies that, in fact, this platform could be used as a diagnostic for Parkinson's disease. So just to say that, in fact, if you didn't know, Parkinson's is way behind Alzheimer's disease. So there's no blood test, there's no PET imaging probe. It's very difficult to diagnose Parkinson's disease. It only happens when people have severe symptoms, and at that point, 60% of dopaminergic neurons, 60 to 80% of dopaminergic neurons are gone, degenerated. It's very, very late. So it's very hard to also develop a therapy at that point. So there's a lot of work obviously happening here. And the main type of biomarkers that people are looking at is mitochondrial dysfunction, oxidative stress, and obviously protein acclimation, phosphosynuclein, synuclein aggregation. Obviously, DJ-1 is very much relevant to oxidative stress and mitochondrial dysfunction. Now, the question is this really specific to Parkinson's? Because as I mentioned to you, the DJ-1 is involved in diabetes, male infertility, cancer, in fact, even in sepsis. So maybe this is a lot more complicated, and it is a lot more complicated, but we are looking at, obviously, we got programs going on in diabetes, Parkinson's, very much also in male infertility. We'll be looking also into cancer soon. But what we are imagining from a business point of view, which is also important if you're sitting in a company, that we may actually use this as a first a general screen, because in the wellness market, this is also very useful. You're 50 years old, like me. I would want to do a redox DJ-1 ELISA screen to see how my oxidative stress levels are. And maybe you get a fingerprint from our assays, and you can say, well, from depending on how your ratios come up, that could be indicative of certain diseases. I'm not going to go into this. So this is what we are trying to do with this, other than really use this as a precision, help us guide our essentially patient selection as precision therapeutic approach. So hopefully with all these, I have given you enough thought about believing that we could actually build a couple of therapeutic candidates, and that DJ-1 is actually an exciting target for Parkinson's and a number of other potentially oxidative stress-relevant diseases. We're thinking of this as a precision therapeutic platform for multiple diseases, obviously focused on Parkinson's first, but we're very interested in, for example, diabetic kidney disease from a glyoxal stress, antioxidant stress point of view, oncology from an inhibition point of view, and actually, inflammation and a number of age-related other diseases, which I won't go into that. So this is kind of talking a bit more about business. This is how we look at this. So we got a precision therapy approach, mostly for Parkinson's disease. Again, interesting in diabetic kidney disease. There's a lot of angles on Alzheimer's disease, which I haven't talked about. Our clinical, this platform, this redox DJ-1 protein ELISA system sits right in the middle of this. It enables essentially our cancer therapy development and on the other side, development of these types of approaches. So with that, I want to thank, I've shown you a lot of data, and thank you for your attention, but I really want to just highlight that I'm just a messenger, and I had a lot of help with this, and we've been at this for since 2010, really. So currently, a lot of the science is run by Balázs Herbert. He's on the biology side, Balázs Flores, biochemistry and biophysics side. Want to really highlight Max, who actually started, I met him in 2010 here in the chemistry department. He was a PhD student, and we have been really working together since then. So he was a scientist first, and now he's really helping and leading fundraising and a lot of business activities at the company. He's also on the board. Also in the audience is Tom Sawyer, who without him we would have never gotten started, and helped raise seed funding, and has been also working in corporate development business and supporting us for long term. Johnny has also a Cambridge PhD. He runs a family office in Hong Kong and Singapore. There's many other people here. I see in the audience Professor Franklin Agbirio, who I've also been working for more than 10 years. He's been also very supportive as a scientific advisory board member. And then Professor Peter St. George Hislop, he used to be, I believe he's no longer a professor here. He's now at Columbia. Chris Roche at Purdue University, Lisa McConaughey at UCSF. So these researchers has fundamentally supported my work and our company's work. And then this is a bit of older picture of the team here. And just to say, there's many other people that have contributed, many different organizations, and this is probably half of the people that on the science labs have been working on this. So with that, thank you for your attention, and I'm happy to receive questions and try to answer them.
Thank you so much for this very insightful presentation. So now the floor is open for the audience for questions. If you have any questions, please raise your hand and we're going to throw this catch box to you.
Okay, so does it work? Apparently, yes.
Thank you very much. Very interesting talk. Thank you. I'm curious about the diseases that DJ-1 is involved in. Is there any comorbidity of those diseases, or the defect that the defective DJ-1 or underexpressed DJ-1 depend on the compartment of the body, and then how does that then implicate the diagnostic tools that you might be developing? Do you need then like a sample from different areas of the body, or how does this relate? Okay. Yeah. There's a number of diseases where DJ-1 have been implicated, and some of these implications actually are underscored by studies showing changes in DJ-1 levels across specific cell types and tissues. And I think the critical issue here is that, and it goes back to possibly oxidative stress and redox homeostasis. There are certain organs and certain cell types that are more sensitive to oxidative stress, and essentially, if you get your redox homeostasis or antioxidant system damaged, then they're more sensitive to a bit of the oxidative stress increase, ends up essentially causing more damage. And there are some cell types that are just a lot more, less sensitive to this. And I think those, and it goes back, those are in those organs or those cell types, you got cell damage and leading to very specific diseases. So when you build your diagnostic tool, would you then need to tailor it for different cell types you're going to be? Yeah. So, that's the big question, right? So the question is, like in Parkinson's, something's going wrong in the brain. Will you see that in the blood? And I think that has been the billion-dollar question also in Alzheimer's disease, right? And what happened in Alzheimer's disease is that in fact, some of the phosphosynuclein, phospho-tau, for example, leaked out and can be detected in plasma. Now, the big breakthrough there, everybody knew that you should be looking at phospho-tau or some pathological modifications that are relevant. But what happened there is that some ultrasensitive detection methodologies, were developed that enabled really the detection, because these are in very low levels. Now, with DJ-1, it's a bit different because we got DJ-1 is at low levels, but not as low levels as some of the A beta or phospho-tau. So we have a sensitive ELISA system, but we don't need ultrasensitive technologies to detect it, which is good because it can be very cost effective. Now, in Parkinson's, we don't know, right? So clearly, there's a lot of DJ-1 coming from not just the brain, but from everything else. So when we do our clinical cohorts in Parkinson's, we don't know what's going to happen. But there's a lot of implications that Parkinson's is not just a brain disease, because you have also peripheral symptoms very early on. Okay, so I think I'm hopeful that we'll see something. But we don't know, and this is the big challenge. That's why there's no blood test for Parkinson's, because we haven't, people haven't been able to identify the marker that actually is specific in the blood. That's the hard part. I'll give you another example: male infertility. So there, in the sperm, you have high oxidative stress, and issues. So there it's a perfect, because we can do a sperm test, we can lyse the sperm, and we can actually see what happens inside, and we can have a good diagnosis. So that's kind of a liquid biopsy, and so that's easy compared to the brain. So that's why we're also looking at, from a business point of view, very keen on looking at male infertility. And by the male infertility is a huge problem. It's a huge also market. So yeah, long answer. Yeah.
Hi, that was an interesting talk. Most of the data that you showed with cell-penetrating DJ-1 seemed to show that DJ-1 is a good prophylactic molecule. You always had it either before nucleation or before you stress the cells. The real question is whether it would be a therapeutic. So when the cells have already got the inclusion, are already dying, will it rescue the cell? And I didn't see any data in that regard. So is this just a prophylactic molecule, or is this going to be a therapeutic tool for neurodegeneration?
I think that, well, the way you put it, I think it's still a therapeutic or a preventive, right? Which is very important.
But you don't take it before the onset of disease, right? That's the difference.
Yeah, okay. Well, I think just to kind of step back, right? So our aim is not to rescue dying neurons. Okay, that's not our aim, because that's too late. If someone's dying, it's going to be very difficult to stop that. We want to prevent it. So if you look at it, as I mentioned, you have symptoms in Parkinson's after you lost 60% of your substantia nigra. So what that means that you're still quite functionable with half of your substantia, dopaminergic neurons around. So if you know this is happening and you can slow down disease progression, then you're fine, right? So, and that's the easier one. What you're trying to do, what you're saying is, I think it's much harder. So we want to do the easy things first, which is not so easy. About rescuing. So as I mentioned, actually DJ-1, there are some studies suggest that DJ-1 is involved in cell survival pathways. So we could actually maybe turn off apoptosis signaling or send a signal for cell survival. So that's also possible. We haven't looked into that, and yeah, we haven't done those models. And you're right, we haven't shown. So we're really looking at this as a preventive. We've been looking at preventive because that's the easier. These are easier models, much easier models than what you're talking about. But I think I want to highlight that remember, there's no proliferative or preventive. So this is also a big value at this point, I think.
Have you thought about the practicalities of delivery and how much of the protein would you have to deliver? And are you going to deliver it directly into the brain? Will it then stay localized to the brain, or do you want it to cross the blood-brain barrier?
Yeah, so there's a lot of challenges in delivering proteins to the brain, and for neurodegenerative diseases, there are a number of companies that are taking different approaches. I probably cannot answer a lot of your questions, and also we have a lot of data that I cannot tell you now because, but I'll give you a couple of examples. So we're looking at different administration routes. One interesting administration route is nasal delivery, because that would straight go into the brain. So there are some technologies there that we're looking at. I haven't shown any in vivo PK data, but if you do an IP injection, then we have very nice distribution, so it does get distributed. Brain access is an issue. There's limited brain access, as you can imagine. So I think there's a lot of questions that we're trying to do, and there's a lot of tricks we are also looking at, which I cannot disclose, about levels and all that stuff. But these are actually, you have very good questions, and these are actually the challenges that we are trying to mitigate and get around.
And if you get it into the brain, can you use the blood-brain barrier as an advantage so it doesn't cross into the rest of the body and reduce on-target toxicity?
Yeah, I mean, that's also another question, a good question, because as I mentioned, you increase DJ-1 levels, maybe you cause cancer. So the idea here is not to overexpress DJ-1, but just to supplement missing levels. Okay, and so there's a lot of questions. Yeah.
I'll just a completely speculative question. Would it make us live longer, healthier, if we? I mean, ultimately, that's all we want, right? I'd like to be fixed when I have a disease, but ultimately, I'd rather live old without diseases.
Yeah, look, I mean, the longevity question, obviously we haven't looked at, and there's not that many studies that looked at DJ-1's role in longevity. As I implied, essentially, oxidative stress, you're going back 50, 80 years. there's been oxystress, the theory of oxystress and aging, that oxystress is responsible for aging. We know that now it's a lot more complex, but if you look at a lot of biological processes that are highlighted as kind of driving aging, oxystress is beneath them. Oxystress, in some ways, is a biochemical phenomenon, and a lot of times, people look at biological phenomena. So I'll give you an example. Mitochondrial dysfunction is one of the drivers of aging. But mitochondrial dysfunction, what is mitochondrial dysfunction? Well, actually, mitochondrial dysfunction causes oxystress. Oxystress causes mitochondrial dysfunction. So it's very much underneath. So it could. So of course, if you're an investor, I'll say yes, sure, it could. But as a scientist, I can say we don't have the proof that it does. We're looking at this from obviously as a therapeutic, not from a longevity point of view. I would love to look at it from a longevity point of view. It's pretty hard to get funding for that.
All right. I apologize that I'm afraid it's all the time we got for the Q&A. So please join me in another round of applause for our speaker tonight.
Thank you.